Structure of DNA and RNA. So what are the components of DNA? How are these components arranged within the DNA double helix? What is the function of DNA? So just to see where this topic comes in the biological molecules overview. So you've got nucleic acids are important information carrying molecules, and then we've got the structure of DNA and RNA. And here is the specification reference for AQA. So you need to know that deoxyribonucleic acids and ribonucleic acids are important information carrying molecules in all living cells. DNA holds genetic information and RNA transfers genetic information from DNA to the ribosomes. Ribosomes are formed from RNA and proteins. Both DNA and RNA are polymers of nucleotides, and each nucleotide is formed from a pentose, a nitrogen-containing base, and a phosphate group. So here is the structure of one nucleotide, and the components of DNA nucleotide are deoxyribose, a phosphate group, and one of the organic bases, adenine, cytosine, guanine, or thymine. And students can also use incomplete information about the frequency of bases on DNA strands to find the frequency of other bases. So, as an introduction, deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA, are important information carrying molecules. In all living cells, DNA holds genetic information and RNA transfers genetic information from the DNA to the ribosomes. So ribosomes are formed from RNA and proteins, and ribosomes are obviously the place in which proteins are made or manufactured. So ribosomes are formed from RNA and proteins. Both DNA and RNA are polymers of nucleotides. So each nucleotide is the monomer which makes up the polymer. And each nucleotide is formed from a pentose sugar, so 5-carbon sugar, a nitrogen containing organic base, so adenine, thymine, cytosine, or guanine for DNA. In RNA, the thymine is replaced with uracil. So you've got the pentose sugar, you've got a nitrogen containing base, and you've got a phosphate group. So looking now at the structure of DNA. So DNA is a double helix structure of deoxyribonucleic acid, and it is immediately recognisable because of its double helix structure. So DNA is a chemical which determines the inherited characteristics, and it contains information, the genetic code, and it passes features from generation to generation. Discovery of the molecular arrangement of DNA is remarkable despite its complex structure, and DNA is made up of 3 basic components from a nucleotide. So DNA polymer is made up of subunits called nucleotides, and it's got 3 components. So for DNA, the sugar is deoxyribose. You've got the phosphate group, and you've got the organic bases adenine, thymine, cytosine, or guanine. So cytosine and thymine are single-ringed, and adenine and guanine are double-ringed. And the 3 components combine by condensation reactions to give a mononucleotide. And then many nucleotides join together by condensation reactions to form polynucleotides. So in 1953, Watson and Crick found out about the structure of DNA. They found that DNA is made of 2 strands of polynucleotides, and they are joined together by hydrogen bonds between certain bases. So the DNA is like a ladder where phosphate and deoxyribose form the sides, and the base pairs form the rungs of the ladder. And a condensation reaction between 2 nucleotides forms a phosphodiester bond. Okay, so you can see here that if you've got one nucleotide here, to keep on repeating this by condensation reaction, you have to join them by making a phosphodiester bond. So each of these individual ones is an ester bond, but when you join them together by condensation reaction, you form a phosphodiester bond, which forms a sugar phosphate backbone. Another thing to point out is that the strands go anti-parallel, they run in opposite directions. And just when we get onto base pairing, you'll see that there are 3 hydrogen bonds between cytosine and guanine and 2 hydrogen bonds between adenine and thymine. So the way in which they pair up is by complementary base pairing. So because the single rings are shorter and the double rings are longer, for it to be the same length, you must have a double ring combining with a single ring. So the way in which they pair up are adenine pairs with thymine and cytosine pairs with guanine, and the hydrogen bonds form between them, between the complementary base pairs. So the quantities of A and T and the quantities of C and G are always the same, but you might have different proportions of each in each species. And just to reiterate that there's 2 hydrogen bonds between adenine and thymine, but there's 3 hydrogen bonds between guanine and cytosine. So we've said about the double helix structure, the fact it's got a ladder-like arrangement of the 2 polynucleotide chains, but then that becomes twisted to form a double helix. And the sides of the phosphate and the deoxyribose wind round each other to form this double helix, and they form the structural backbone of the DNA molecule. And for each complete turn of the helix, there are 10 base pairs. Okay, now looking at the RNA structure, the mononucleotide is made up of, instead of deoxyribose sugar, because we're looking at RNA, it's actually a ribose sugar. So in RNA, the sugar is ribose. You've still got the phosphate group, but then instead of thymine, you've actually got uracil. So that should actually say uracil there. So you've got cytosine and uracil, which are the double rings, and adenine and guanine. So you don't have thymine in RNA. It's instead replaced by uracil. So an RNA molecule is a relatively short polynucleotide single-stranded chain. So it's single-stranded, it's made up of a ribose sugar, and instead of thymine, it's got uracil. So what is the function of DNA? Well, DNA is a hereditary material which passes genetic information from cell to cell, from generation to generation, and there are 3.2 billion base pairs in DNA of a mammalian cell. So there's infinite variety of sequences of bases along the length of the DNA molecule, and this variety provides genetic diversity of the organisms. And it is adaptive for its function because it is stable, so it can pass from generation to generation without changing. The 2 separate strands, which are joined by hydrogen bonds, allows them to separate during DNA replication and protein synthesis. It's a large molecule which carries lots of genetic information. It base pairs with helix of the deoxyribose backbone. There are base pairs within the helix of the deoxyribose backbone, and the genetic information is protected from the outside chemicals. And the sequence of bases is very important in everything it does. There's just a few definitions. So complementary base pairing is the phenomenon where guanine always hydrogen bonds to cytosine and adenine always binds to thymine. And the bond between guanine and cytosine shares 3 hydrogen bonds compared to adenine and thymine, which always share 2 hydrogen bonds. And the complementary base pairs of guanine with cytosine and adenine with thymine connect to one another by hydrogen bonds. So there are 3 hydrogen bonds between C and G and 2 hydrogen bonds between A and T. And these hydrogen bonds between the complementary nucleotides are what keeps the 2 strands of the DNA helix together. So for now we just look at a worked example. So DNA is a polymer of nucleotides. Each nucleotide contains an organic base. Explain how the organic bases help to stabilize the structure of DNA. So hydrogen bonds between the base pairs holds the 2 strands together, and because there's many hydrogen bonds, it provides strength. Okay, next one. Figure 1 shows a short section of a DNA molecule. So what is R? Well, R is a deoxyribose sugar, and we know this because it says it's a DNA molecule. And Q is the phosphate group. Name the bond that holds A and B together. Well, A and B are bases, so it must be hydrogen bonds. Just a bit of a look at DNA and RNA now. So DNA is a very stable structure. It's found in the nucleus and little bits in mitochondria. It's got a double helix structure. It's relatively long, can be 250 million nucleotide pairs long. The nitrogen bases are adenine, thymine, cytosine and guanine. And its pentose sugar is deoxyribose. And each nucleotide contains phosphate, pentose sugar, which is deoxyribose, the nitrogen base, which can be A, T, C or G, and it's got 2 strands. So mRNA, which is a type of RNA, is a relatively unstable structure. It's found either in the nucleus, the cytoplasm or the ribosomes, depending on where along its journey it is. It's single-stranded, it's relatively short, and the nitrogen bases are adenine, cytosine and guanine. And instead of the thymine, the 4th base is uracil. The pentose is ribose, and each nucleotide contains a phosphate, the pentose sugar ribose, and the nitrogenous base, which could be A, U, C or G, and it is single-stranded. Then finally, another type of RNA that you learn about is tRNA. This is a relatively stable structure because of the hydrogen bonds when it folds in on itself. It is found in the cellular cytoplasm and it's involved in protein synthesis. And transfer RNA brings or transfers the amino acid to the ribosome. And it's got a distinctive folded structure with 3 hairpin loops that form the shape of a 3-leafed clover. And one of these hairpin loops contains a sequence called the anticodon. It is the shortest of all 3, and the bases are adenine, cytosine and guanine, and also uracil. The pentose sugar is ribose, and each nucleotide contains phosphate, pentose sugar, which is ribose, and the nitrogenous base A, U, C or G, and it is single-stranded. So in summary, deoxyribonucleic acid and ribonucleic acid are important information-carrying molecules. In all living cells, DNA holds genetic information and RNA transfers genetic information from DNA to the ribosomes. Ribosomes are formed from RNA and proteins. Both DNA and RNA are polymers of nucleotides, and each nucleotide is formed from a pentose sugar, a nitrogen-containing base, and a phosphate group. The components of a DNA nucleotide are the deoxyribose sugar, phosphate group, and one of the bases, either adenine, cytosine, guanine, or thymine. The components of an RNA nucleotide are ribose, a phosphate group, and one of the organic bases, adenine, cytosine, guanine, or uracil. A condensation reaction between 2 nucleotides forms a phosphodiester bond. A DNA molecule is a double helix with 2 polynucleotide strands, held together by hydrogen bonds between specific complementary base pairs. An RNA molecule is a relatively short polynucleotide chain. You should also appreciate that the relative simplicity of DNA led many scientists to doubt that it carries a genetic code.